Hi, and welcome to the first video in conducting EEG research series. In this video, we'll look at the components that make up the EEG kit and learn how to put them together for data collection. We'll start with a general recap of the theory behind EEG, after which we'll move on to a hands-on tutorial specific to the system we have in the lab. So without further ado, let's get started. First, let's take a quick look at EEG and talk about how it works. EEG, or electroencephalography, is a technique used to measure the electrical activity of the brain. At its very basic, it involves placing small electrodes on the surface of the scalp, allowing us to pick up the electrical signals produced by the brain. Naturally, these signals are very weak and need a little boost, so from the electrodes, the electrical signal first needs to travel to the amplifier. The amplifier increases the power of the signal and converts it to a digital format that can then be interpreted by a computer, which finally records the signal and visualizes it in real time. Without going into too much detail, it's important to understand the type of signal that gets recorded in EEG. Rather than recording the flow of electricity itself, EEG measures the pressure that pushes the electrical flow from one place to another. This pressure is called voltage or electrical potential because it represents the potential of electricity to flow from one place to another and not the electricity itself. So placing a single electrode on the scalp and plugging it into the amplifier wouldn't give you EEG data. Remember, you can only record voltage between two sites on the skin. So if you're interested in measuring activity at a hypothetical site A, what you need to do, aside from placing a so-called active electrode in that spot, is introduce another electrode at a different place on the skin called the ground. Once connected to the amplifier, any activity measured with the setup will represent the flow of electricity between the active and ground electrodes. Unfortunately, the ground electrode will also pick up the electrical noise generated by the amplifier itself. To solve this issue, we'll have to add a third electrode to the setup, called the reference. The amplifier will then record the voltage between active and ground electrodes, as well as the voltage between reference and ground electrodes. The latter will be subtracted from the former, eliminating the ground electrode noise in the process. The measure we then end up with effectively represents the voltage or the potential of electricity to flow between the active and reference electrodes. And that's the EEG data that you see on the screen. In summary, to record EEG, your setup must include at least three types of electrodes: active, ground, and reference, which are typically embedded in a single cap, as well as the amplifier and the computer with specialized software. Together, these components make up what is known as the data acquisition system. EEG can be supplemented with recordings of other types of electrophysiological activity, such as EOG for the eyes, EMG for the muscles, and ECG for the heart. Much like EEG, these recordings are obtained with electrodes placed on the skin in specific locations. These auxiliary electrodes can be plugged into the amplifier along with the EEG cap to produce a combined electrophysiological recording. It should be noted that some EEG caps come with one or more EOG electrodes already built in which case you don't need to plug anything extra into the amplifier to record ocular activity. As I said earlier, this setup really has all you need to collect brain activity data. But as researchers, we're not just interested in recording what the brain is doing, but also when and why it's doing it. For that reason, EEG data is often collected while the participant is engaged in some sort of task, like counting the occurrences of a stimulus or pressing a button every time they feel a certain emotion. In these cases, it's especially important to record precise timestamps of when the events of interest had occurred. This means that our setup requires the addition of another computer, which will present the stimuli to the participant and in some cases collect their behavioral responses. Note that ideally, this should be a separate computer, not the one that's running the data acquisition software. Running any other programs in parallel with that software can lead to timing issues and even data loss, so it's better not to risk it. We then also need a way of adding this data to our EEG stream so that the timing of events is synced with the brain response. This can be done by plugging the stimulus presentation computer into the amplifier using a special converter. The amplifier will then combine that information with the rest of the signal, and the data acquisition computer will display it as timestamps in the software. The stimulus presentation computer and the converter it uses to connect to the amplifier make up the stimulus presentation system. As a side note, if you're not using any computer-generated stimuli and you're more interested in, say, the participant's response to their immediate environment, you can use a response device that plugs into the amplifier in the exact same way. And every time the response is given, the data acquisition software will record it as a timestamp. So now that we're familiar with all the different components that make up an EEG kit, let's move on to the specific kit we have in the lab. To record EEG, this lab uses A&T Neuro's EEG Sports solution. It includes the EEG Sports amplifiers, the waveguard connect caps, and the EEG 64 recording software, which is currently installed on the Surface Pro 9 tablets. The solution also includes a bunch of accessories, which we'll cover later in this video. For now, let's take a closer look at the core components of our data acquisition system. So first, the EEG Sports amplifiers. These are pretty lightweight and have a battery life of up to 5 hours, making them suitable for both stationary and mobile on-the-go recordings. At the bottom, each amplifier has 2 ports for plugging in the caps, labeled 1 and 2. At the top, we have the power on and off button, a port for plugging in additional electrodes labeled bipolar in, and another port for plugging in any trigger devices. Finally, each amplifier has a USB port for connecting it to the data acquisition computer and a charging port. Each amplifier comes with its own charger. It's very important that the amplifiers are charged regularly, even when they're not in active use. Letting them run out of power can actually damage the batteries. That said, it's also not a good idea to leave them charging for days on end, so ideally you should charge them for a few hours every week while you're in the lab and unplug them before leaving. Moving on to the waveguard connect caps. These are gel caps, meaning that they require the use of conductive gel in order to establish connection between the electrodes and the scalp. The caps come in 3 sizes, small, medium, and large, which are made for head circumferences of 47 to 51, 51 to 56, and 56 to 61 centimeters respectively. The caps use pure tin electrodes that work well with saline-based gels or pastes. The specific caps shown here are a modified version of the standard model, specially made to include 4 drop-down EOG and 2 mastoid electrodes, which can be taped to skin during data collection. On top of that, the caps have 61 EEG electrodes, which are embedded in the cap itself and positioned according to the extended 1020 system. By default, the AFZ electrode acts as the ground and the CPZ electrode as reference. The reference site, however, is subject to change. It can be changed both in the recording software before you start the recording or once the data has already been collected during analysis. The caps have 2 shielded cables, labeled 1 and 2. As we'll see later in this video, these numbers correspond to the labels on the amplifier's 2 EEG ports and are here to help you match the right input to the right output. Finally, let's take a look at the data acquisition software. As mentioned earlier, it's currently installed on the Surface Pro 9 tablets, and unfortunately, it can't be installed on any additional devices due to licensing. The license can be transferred to a different device, but it's not an easy task and will require help from the A&T Neuro customer support as well as the university IT. Both tablets are password protected and share the same user account specific to this lab. You shouldn't try to log in using your personal university details. The username and password for the lab account will be shared with you before you start working with the kit. The EEG 64 software can be launched from the desktop when the tablets are connected to EduRoam, the university Wi-Fi network. If you try to launch the app without Wi-Fi connection, you'll just get an error. To connect the tablets to EduRoam, you should follow the standard procedure outlined in the IT support section on the university website and use your personal university account details to set it up. When you successfully open the EEG 64 app, you'll see the home screen. Of course, it might look slightly different from what I'm showing here if you're using a newer version of the app. In the top left, you'll find tabs that allow you to switch between different modes or workflows: acquire, review and export, and a few other options. To record EEG data, you're going to be using the acquire workflow, which we'll explore in detail further on in the series. Now that we've covered the core components of the data acquisition system, let's move on to stimulus presentation. As we've seen earlier, this consists of a computer and a converter. The converters we use here in the lab are the USB to TTL8 adapters by lab hackers. On the one end, they have the USB Type A plugs that go into the computer, and on the other, DB25 plugs for serial connection with the amplifier. In our specific setup, the serial plugs also have the gender changers attached to them. As for the computer, this can in theory be any device, whether it's your personal laptop or something we already have in the lab. Of course, the specs of this device will affect the performance, quality, and timing accuracy, so there are certain rules of thumb when it comes to choosing a stimulus presentation machine. First of all, it's always best to avoid using MacBooks or computers running macOS in general. Second, you'll want a device with a good graphics card, fast refresh rate, and a decent RAM, so something like a gaming laptop would be ideal. This shouldn't matter too much if your experiment involves simple visual or auditory stimuli, but if you're planning on showing large video or image files or collecting data through some interactive game-like paradigms, the computer specs are definitely important to consider. To program experimental tasks, in this lab we usually use SciCopy, a free Python-based software package for designing and running experiments. SciCopy provides both a user-friendly graphical interface and a basic code editor, which makes it a good choice for beginners and advanced users alike. We have a separate video called Introduction to SciCopy where we cover the software in great detail, so I highly recommend checking it out. The important thing to note about SciCopy is that it allows to send stimulus-specific timestamps, also known as triggers or event codes, through the USB to TTL8 adapter and into the amplifier. These have to be numeric and formatted in a very specific way outlined in detail on LabHacker's website. We can also use special triggers to start and stop the EEG recording, which requires some extra configuring on the data acquisition side. Of course, as long as you know what you're doing, you don't have to use SciCopy. The USB to TTL8 adapters work with Python as well as MATLAB. But if you're just starting out, SciCopy is probably your best choice. So having looked at both the stimulus presentation and the data acquisition gear, it's time to put it all together. First, we'll connect the stimulus presentation computer to the amplifier. The USB end of the LabHacker's adapter will go into one of the laptop ports, and the DB25 connector will be attached to a trigger adapter labeled XS230, which was included with the amplifier. The other end of the trigger adapter will now go into the amplifier port labeled trigger. And that's the stimulus presentation side of things. As for data acquisition, we'll now have to connect the EEG cap to the amplifier. The ports for the EEG caps can be found on the opposite side of the amplifier body, labeled 2 on the left and 1 on the right. We'll take the 2 connectors attached to the cap and plug them in, making sure that cable 1 goes into port 1 and cable 2 goes into port 2. Finally, we'll connect the amplifier to the data acquisition computer with a USB cable, plugging the Type B side into the amplifier and the USB Type C into the Surface tablet. We can now launch the EEG64 app on the Surface and turn on the amplifier with a simple press of the power button. If the amplifier is charged and ready to record, its LED indicator will glow green. We will also launch our stimulus presentation software, which in this case is SciCopy, and get the experiment file ready to run. With that, our minimal setup is complete and ready for data collection. The next steps will involve working our way through EEG64, which is something we'll do in part 2 of the series. To end this video, I'd like to introduce a few add-ons and accessories that can be used with the main kit to extend its functionality. Note that the section is meant to give you a broad idea of the types of things you can do with the kit, so I won't be covering any of the devices in detail. It's important that you do your own research if you're planning on using any of them in your studies. So first, we can add more sensors to our data stream to record additional physiological or environmental signals. To do that, we need to connect a little extension device called the sense box to our amplifier. Each sense box can accommodate up to 6 additional channels. These channels can be either bipolar with 4-pin connectors or auxiliary with 5-pin connectors. The sense box also has a port for its own ground electrode with a single pin. To add sense box to your setup, start by plugging it into the bipolar port of the amplifier. Then take an Anchor power bank and plug it into the sense box with a USB cable. This step is super important since the sense box doesn't have an internal power source. Now that it's connected to the amplifier and the battery, you can choose to plug in one or more sensors. You can record temperature with a temperature sensor, galvanic skin response with the GSR sensor, respiration with a respiration sensor, movement with the accelerometer, and intensity of light with the photodiode. The latter can be quite handy for testing the timing accuracy of your setup or even for synchronizing your visual stimuli with the EEG stream. Note that all of these sensors make use of the 5-pin auxiliary connectors. Additionally, you can record ECG with the combination of the double-ended adapter cable and the disposable ECG electrodes that are glued directly on the skin. Unlike the rest of the sensors, these electrodes must be connected to the 4-pin bipolar ports. Being bipolar, these electrodes record the electrical potential between 2 adjacent sites on the skin where they are placed. Note that adding any sensors to your setup requires some software reconfiguring, which we'll cover in the next video. Next, I'll introduce an alternative input device, the response button, that serves to mark the EEG data whenever a participant presses it. To add it to your setup, you'll need to connect the cable end labeled 1 to the XS230 trigger adapter before plugging the adapter into the trigger port of the amplifier. You can either use the button on its own or combine it with the stimulus presentation system triggers by connecting the USB to TTL8 adapter to the cable labeled 2. Finally, if you're interested in doing mobile recordings where the participant is moving around while their EEG is recorded, you'll have to make some changes to the setup, and most importantly, introduce the backpacks. These are designed for the participant to carry the data acquisition system behind their back with special pockets for the amplifier, the Surface tablet, and the sense box if it's being used. Once all the components are secured in their pockets, the backpack can be worn by the participant both indoors and outdoors. That said, you should always watch out for the rain when recording outside, since the backpacks are not waterproof, and apart from the caps, the system should never come in contact with water. You'll notice that the backpacks can only fit a single tablet at a time, so if you want to present any stimuli, you'll either have to come up with a way to carry the stimulus presentation system separately or do the unthinkable. That is, get rid of the stimulus presentation system and run the experimental tasks on the data acquisition device. To sum up, in this video we've briefly looked at the theory behind recording EEG and covered the various components that make up our EEG kit. In the next part of the series, we'll take a closer look at the data collection software and learn how to set it up for your experiment. Also, in future episodes, I'll demonstrate how to cap participants using the WaveGuard Connect gel caps, record the data, and eventually export and analyze it. Hope to see you in the next video. Bye for now.